Hi, my name is Harald Schmidt. I'm the coordinator of the REPO trial project. And this is the final instruction video on the concept of in silico trials and using a completely new way of defining diseases, which then results in a much more precise mechanism-based disease definition, diagnosis, and most importantly, therapy. So the two validation trials that we conducted at the end of REPO trial was a trial in ischemic stroke named REPO stroke, and it was a phase 2 trial, so proof of concept in patients. And the other one was named REPO HFpEF, also a phase 2 trial, in a specific form of heart failure with preserved ejection fraction. Stroke is a condition that is apart from removing the obstructing thrombus, either by dissolving it or by physically removing it, there's no therapy. And heart failure with preserved ejection fraction is a form of heart failure where the objective criterion, namely which percentage of blood in the left chamber of the heart is actually ejected per heartbeat, is technically normal, but the patients still have heart failure. And this form of heart failure does not respond to the typical drugs that are used for other patients with heart failure, where the ejection fraction is actually reduced. So two highly unmet medical need conditions. And we were collaborating here with the university clinics in Essen, Benedikt Frank, and with respect to the heart failure trial with Daniel Setting from the University of Halle. And the trial was eventually conducted at the University Clinics of Valencia by Julio Núñez. To start with, I have to recapitulate a little bit again where we came from, namely from the network of all human diseases. Here, the diseases are linked through common risk genes, and we are working on this cluster of diseases which share obviously a lot of risk genes and therefore may have a similar mechanism. When we zoom in, we see stroke and heart failure as some of the disease phenotypes or symptoms that we find here, and they share a lot of risk genes. And not only are they connected through common genes, but also the diseases occur very often in the same patient, so comorbidity, and they share a lot of symptoms. So obviously these diseases have a lot in common. And so we thought they may also share similar molecular mechanism and maybe treated by similar compounds. Some of the genes that were found here are related to cyclic GMP signaling, others to reactive oxygen species, metabolism, or pathway. Normally, geneticists annotate them to these maps of pathways. However, we think that these pathways are not really very useful because you have here about 300 proteins, and which one would you possibly use for a diagnosis and a treatment now? So what we did, we used those genes and constructed the signaling module, as you see in the other previous webinars in more detail. De novo basically using the risk genes as seed proteins looking for the first neighbor. Then we found this module. We used different methods, and they all found that there is apparently an NADPH oxidase free radical forming enzyme interfering with cyclic GMP signaling. So in the end, from these two rather large pathways, only those two little bits were relevant and apparently form, at least for some of the patients, the relevant signaling module. This is not only a mathematical or theoretical construct. It has recently been shown that indeed in our cells we have little protein communities which have actually the size of 10 to 20 proteins. They are interconnected. So this is actually how signaling works, and such little nanomodules where signaling happens and which are interconnected has also been found by cell staining immunofluorescence research. Now, what's actually going wrong in heart failure with preserved ejection fraction, and actually also in hypertension in some patients, is that this cyclic GMP signaling pathway and formation of reactive oxygen species normally run in parallel, not interfering much with each other, but it can happen that some patients have, for instance, too much of this enzyme, then too much of this reactive oxygen species is formed or at the wrong site, and then it interferes with this signaling pathway, which, for instance, causes vasodilation, especially of small blood vessels, which supply, for instance, the heart muscle with oxygen and nutrients. And of course, if this is impaired, then the heart muscle doesn't function very well, and we postulate that this is one cause of heart failure with preserved ejection fraction. So there are a couple of drugs that we can use to treat this condition. This enzyme here that forms the signaling pathway is damaged in heart failure with preserved ejection fraction, but it can be activated through another compound. Even the damaged form of the enzyme can be activated. So we have normal formation of this messenger again, cyclic GMP. And this enzyme up here is also damaged, but this can be sort of recoupled by this folic acid, a vitamin, and by offering this amino acid, which is then converted to arginine, the substrate for NO formation. And we therefore combine this SGC stimulator, folic acid, and an amino acid, a triple therapy, which then attacks at several sites and more or less normalizes this signaling module again. The trial was conducted in a way that we did a 2-to-1 randomization. Patients either received triple therapy or the best medical care for comparison. This shows the different visits. This shows the combination with the citrulline and folate, and then we up-titrated the SGC stimulator, and there was a follow-up phase of 30 days where patients were interviewed. So we have fairly acute data, but we also have long-term data, whether the patients benefited from this treatment. Importantly, we do not treat patients all the same. In stroke, for instance, there is the same module affected, but in a different way. Here, we have the occlusion of a blood vessel that supplies the brain with oxygen. And under these conditions, another enzyme gets induced and produces much larger amounts of reactive oxygen species as just the previous enzyme that was upregulated does. And then all of a sudden, we have a highly toxic chemistry, which supersedes in importance almost the deficiency in the cyclic GMP signaling. There are so many free radicals and toxic nitrogen oxide species occurring that are plain cytotoxic. And in this condition, you don't want to recouple endosynthases to basically feed more NO to form more of these nitrogen oxide radicals. You actually want to stop this process here, at least for a couple of hours, but still enable this cyclic GMP signaling, which protects neurons and also closes the blood-brain barrier. That's the separation between the circulating blood and the brain, which protects the brain a little bit. And in stroke, this barrier becomes or leaks. And so we can use again this compound, but to inhibit those enzymes and the endosynthases, there were actually no drugs on the market, but we found that we could use a drug that is used to inhibit the thyroid and an antipsychotic drug, that they also are perfect inhibitors for those two enzymes. So that process is called drug repurposing, and this is what we used in this stroke trial. So combining those two drugs, and again, use this SGC stimulator or activator. Why is this drug repurposing so successful? This figure shows over 400 drugs that are approved in the United States on the market. And here it's measured to how many proteins these drugs actually bind. And a little bit surprising, a drug is not only binding to the protein for which it was actually designed. In average, it binds to 32 other proteins. Some drugs bind only maybe to 1 or 2, but other drugs here in the front bind actually to hundreds of proteins. And the reason why that happens is that these small molecule drugs need to bind to little pockets in proteins. And apparently, the number of different pockets on all our proteins is limited. And these pockets occur again on different proteins. And if such a pocket is present in hundreds of other proteins, that compound will bind to hundreds of other proteins. And if this pocket is pretty rare, okay, then the drug will only bind to one protein. So in this case, we could use the fact that those two compounds actually also bind to this, and this one also to this enzyme, and we conducted the clinical trial with this. The RepoStroke trial had a similar setup. The follow-up was so that we also had a phone interview again, but in addition, imaging. And before that, the randomization was actually 3-1, so a little bit more patients received the therapeutic. The important thing here was to define the treatment interval where it most likely made sense to treat the patients with this combination of 3 drugs, namely then when this toxic mechanism still occurred. Once this process stopped, it would not make sense to give those drugs much longer. So, you're probably wondering when the results will be released. The heart failure trial, we expect to have results in about February 2024. And the stroke trial, we expect to be able to release the results in March 2024. So stay tuned for our website. We will, of course, announce it, hopefully exciting positive results through social media, but also on our website. But the most important thing is that you see how one can convert such a basically in silico design of disease mechanism together with diagnostics or just a defined time window, and using the right drugs that correct such a signaling module directly into a clinical trial, hopefully with positive results. So stay tuned. Thank you very much.